Deep inside a flower's ovary, a microscopic pollen tube breaches the ovule through a narrow opening called the micropyle, delivering a specific genetic payload. Once inside one of the recipient synergid cells, the tube ruptures, releasing exactly 2 male gametes directly into the cytoplasm. This initiates a divergence in development. Most animals only need to construct their own organism during early growth. Flowering plants, however, must build the embryo while simultaneously constructing a separate, isolated metabolic battery to power that embryo later. They achieve this through a phenomenon called double fertilization. 2 gametes trigger 2 distinct, synchronized cellular events inside a single embryo sac, a mechanism found only in flowering plants. Understanding the mechanics of this sequence, from cellular fusion to metabolic dormancy, explains how plants survive environmental extremes and flaunts the basis for how we grow food today. This schematic cross-section maps the terrain of the mature embryo sac. At the micropolar end is the egg apparatus, containing the egg cell. Further up, located in the middle of the large central cell, are 2 distinct polar nuclei. The first male gamete moves toward the egg cell and fuses with its nucleus. This completes syngamy, resulting in a single diploid cell, the zygote. This is the blueprint for the new plant. The second male gamete travels to the central cell and fuses with the 2 polar nuclei, producing a triploid primary endosperm nucleus. Because this secondary event involves the fusion of 3 separate haploid nuclei, it is termed triple fusion. This dual pathway mechanism ensures metabolic resources are only synthesized if a viable embryo is actually formed. It prevents the plant from wasting energy on an unfertilized ovule. Once the zygote forms, it hits a developmental bottleneck. It will not divide or grow further until a sufficient amount of endosperm tissue is constructed around it. To build this energy reserve, the primary endosperm cell divides repeatedly, undergoing successive divisions without immediately forming cell walls, creating a mass of free nuclei. This stage is visible in tender coconut water, which consists of thousands of free nuclei suspended in liquid. Eventually, cell walls form during cellularization, turning that liquid into the solid white kernel. The zygote waits for this tissue to form because it provides the guaranteed nutrition required for the embryo to survive its early development. Fueled by the endosperm, the zygote initiates embryogenesis. It expands from a simple proembryo into a globular shape and eventually into a heart-shaped structure. This comparative blueprint shows the 2 paths. A mature dicotyledonous embryo is structured around a central embryonal axis, flanked by 2 thick cotyledons that store food reserves. The axis above the cotyledons is the epicotyl, terminating in the plumule or shoot tip. The axis below is the hypocotyl, which ends at the radicle, the root tip. Monocots, like the grass family shown on the right, possess only a single cotyledon called the scutellum, which is situated towards one side of the embryonal axis. These embryos feature specialized protective sheaths. The shoot apex is enclosed in a hollow structure called the coleoptile, while the radicle is enclosed in the coleorhiza. Despite these morphological differences, both architectures are optimized to protect the delicate meristematic tips during the physical stress of germination. As the embryo matures, the outer integuments of the ovule harden into a protective seed coat. Seeds are categorized by how they manage their food supply. Non-albuminous seeds, like peas, consume their entire endosperm during development. Albuminous seeds, like wheat, retain a portion of it for use during germination. At this stage, the seed purges its water content down to 10 to 15% by mass. This dehydration slows metabolic activity to a near halt, entering a state of inactivity known as dormancy. While the seed enters dormancy, the surrounding ovary develops into a fruit, with the ovary wall becoming the pericarp. This dormancy enables survival across vast timelines. In the Arctic tundra shown here, researchers recovered a seed of Lupinus arcticus from the permafrost. It successfully germinated and flowered after an estimated 10,000 years of dormancy. A similar record exists for a date palm seed discovered during an archaeological excavation at King Herod's palace. Despite being 2,000 years old, the seed remained viable. By combining physical armor with metabolic suppression, flowering plants convert embryos into biological time capsules, allowing their offspring to survive until conditions are favorable for growth. Although seeds are typically the products of fertilization, certain species, like grasses, have evolved a way to bypass this requirement. This process, called apomixis, allows for the production of seeds without fertilization. This chart contrasts the two pathways. In apomixis, a diploid egg cell is formed without the usual reduction division. It then develops directly into an embryo, bypassing the need for a male gamete. A related phenomenon is polyembryony, frequently seen in citrus, where multiple embryos of different sizes and shapes form within a single seed. This bypass has significant economic potential. Current hybrid crops offer high productivity, but their seeds cannot be saved for the next season because their specific traits segregate and are lost. Farmers must purchase new hybrid seeds annually. This photo of large-scale corn production represents the scale of modern agriculture. If high-yield hybrids could be modified to use apomixis, the resulting seeds would be genetic clones of the parent, allowing farmers to maintain the same productive traits year after year. Research into the genetics of apomixis aims to stabilize these hybrid traits, potentially providing a way to make high-yield agriculture more accessible and sustainable globally.